In this video, I will talk about what is a standard curve. And if you are going to start watching this video, I highly recommend you that you watch first the other two that I already posted. Because in this one, I'm just going to show you examples and a question that they could be in the test. So for example, I want that you remember that standard curves represent a relationship between two quantities. And although here they said a curve, it actually is a line. Your data will start looking at the end like a curve, but you're only going to take a line. And these are also called calibration curves. And if you watch the video, you know why they are called calibration curves. That is very important. And they are used to look for unknown concentrations of different substances. So for example, concentration of glucose, concentration of proteins, as we did in the class. In the video, you can see that they also have DNA concentration. And in future classes, you will see what is NADH. That is very important. So if you have a substance, you can use these calibration curves. So if you know the absorbance of a specific substance, with that absorbance, if you know the concentration, but you have already plotted the data, you can make a line, as you can see here, and know exactly what is the concentration as they show in the video. So remember, important, what is a concentration curve? So in the test, maybe I ask you this question and you need to know that they show the relationship between two quantities. Remember that they are also called calibration curves. Remember that they are used for calculating the quantity of unknown sequence of unknown substance. And for example, how you do it, you just, if you have a chemical, a substance, you just need to calculate the absorbance, and with that you extrapolate here the absorbance and you can get the quantity, the concentration of that substance. As it could be protein as we did in the lab, it could be DNA as you watch in the video, or it could be other substance as you will do in chemistry and in other classes. So again, this will be the test, and if you watch the video, you understand now much better what is a standard curve, a calibration curve. And so for example, this tiny ruler, she gave this example that this tiny ruler is not useful to measure a car because it will be too small. Yeah, you need something, a different type of a different way to measure the entire curve that is much bigger. Something like that happened with the calibration curve. This is why not all of you got perfect marks. And so in this case, in the lab assignment, some of you made the line very long. Even your data start curving here. So here the calibration curve only work up to here. From 0 to here, these type of points are not useful. You cannot continue doing extrapolating from here to here. You will not be able to do this here. That will be a mistake, because in the same way that you have a problem with the ruler, here your ruler is not as big, it's only for this distance. So be very careful in the lab test. If I ask you something that is outside of the curve, you shouldn't take it into account. And again, I gave you several examples of how to use this standard curve, and one of them is the DNA concentration that you watched already the video. Also pay attention to these serial dilutions. I will make an example when you that it will appear a question like that in the lab test. You will have a serial dilution, and then you need to make a standard curve, and from that standard curve you will have to calculate it using, if they give you, for example, the absorbent, in this case it was the CT, you have to calculate the quantity, or in our case it was the concentration of the protein. And then I will explain how to calculate that. And something that is not going to be in the test, but later you will study that in more detail in chemistry. There are cases that definitely your data is linear. However, there will be other times that your data follow other models. In our case, in the test, I'm just going to ask you for the one that we studied, that is the linear, in which you have to reject some of the points here. And something that you will study in more detail in statistics and also in chemistry is that you're doing a linear regression. Here in red, you see the different observations, and remember that our observations, our measurement, they are not always perfect. So the idea that you are not supposed to make the graph like this will not be okay. The idea that you have to make a linear regression and you have to make a line in this case that going between the points, that is the best fitted line, and go and reread the lab manual that in there we explain how to do it. And this could be a question for the test, that is draw the best fitted standard curve. So remember, this is not the best one. Here, if you see, the dots go from here to here, so this is too short. You can still use this part of the graph. In this one, you made the here you have the opposite problem. Here you went too far in the graph. You are not supposed to have this section and this section. And the best one in this case will be this one, because your dots will go from here to here. In the case of our absorbance and concentration that we study, it actually goes to zero, because when you have zero absorbance, you have also zero concentration. In this case, there is no, it doesn't go to the origin. In our case, the one that we did, it actually goes to zero, but it definitely doesn't cover these sections where the graph start curving. So if I ask you one of those questions, you have to pick this one, remember. And again, you already know that this, you shouldn't include this point, so this one will be wrong, and this one will be wrong, this is too long, and will be wrong because this is too short. And here, something that I wanted to remind you is that you need to know your data very well. So for example, here we have in blue the nucleus, here in green you see microtubules, and here in red you can see actin. Yeah. And here, for example, if I want to measure the blues, the nucleus, how bright they are, here I can make a graph with the pixels. So there is a lot of pixels that are black, the regions where there is not the nucleus, and there are a few regions where you have the nucleus, but you have nucleus that we dyed, and they are very bright. Yeah, so they are here, so they saturate the microscope, and they look very bright. And although the graph should be like this, yeah, because they are much brighter. However, the microscope and the software only allow you to have that. So here you get these peaks that are not real. This is exactly something similar that happened with the absorbance and the concentration, that there is a point when the color, the absorbance and the concentration don't have any more a linear relationship, when the machine is saturated the data, and this section of the graph you shouldn't include it. The same thing that happened here, you should only include this section of the graph, and you shouldn't include this one because this one are artifact. The same thing will happen here. So why I want to tell you this? is because every time that you are working with this standard curve, you just have to focus on the region of the standard curve that you can use, not in the part that have a curvature. And of course, as we have been talking all this time, so this part is wrong, so you have to reject this section, and you can only take this part of the lines. And so again, sorry to tell you this so many times, but so you don't have to include this in your graph, okay? And then when you don't include those one in your graph, you can now plot the line. So look, this is what happened when you don't include the line, and this is what happened when you include the dots that they were not necessary. So this is wrong, and you get a completely different. If you are going to try to again extrapolate and get the concentration here, you get a completely different value here. Yeah, you get between 2 and 4. Here you get between 4 and 6, very much closer to 6. So rejecting those points is important, and this is why some of you didn't get 100% in some of your graph, because you had to reject some of the points. Here you can find an example of a question that I can ask you in the test. So please stop the video and try to read it. Okay, now that you read it, there are two key pieces of information. So you know that they give you the absorbance, and you get the protein concentration similar to what we did in the lab. And here there are two pieces of information that are fundamental. One that is a serial dilution of 50-fold, and you have an absorbance of 30 here. So now, what do you think that is the right question? Yeah, what is the right answer? What is the protein content in the sample? So they give you this new sample, and you went to the spectrophotometer, and it give you this measurement. So now you have to guess which one of this one is the right concentration. So again, those two things are very important. Those 2 points are very important, and we are going to use them to answer that question. So first thing, I divided into 3 steps how to answer that question. First thing that you have to remember is that this is a diluted solution, so it's not the original sample, and this is important because you will need to have to do an extra step if it's a diluted solution. So in the beginning, if the absorbance is 0.30, you make a line, and as this is a calibration curve or a standard curve, you go here, and this is you find the concentration that is around 125, is in the middle. So you have your first point, and this will be, if it was the original sample, here you will have, this will be 1 point round, this will be the answer. However, as it's a dilution, you need to multiply this, and so look, so you have to multiply 125 and multiply by 50, because that was the dilution, and you get this value, and then you turn this from milligrams to gram, and is 6.25. So the answer is like this. Remember, 3 steps: check the concentration if this is the original sample or the dilution, check the absorbance, go later and find it in your standard curve, and finally just you have to multiply your concentration, the serial dilution, and then you can do a conversion to the right measurement, to the, if it's milligrams, or if it's grams, if it's milliliter, if it's liters. And yeah, and that's all. And this will be one of the type of questions that I can ask you in the test.